So we have, I do not have the soft copy, but I'm going to get the soft copy also here on the board. But you have only 20 minutes left. We have very few questions. I will start the discussion of all of the questions at 11:30. So use this 20 minutes to attempt the question paper you have received right now. And once after this, we will discuss some of the questions of last class and questions of today's class. Thank you. So I'll start the discussion at 11:30. I'm showing the question for those who might watch it later. I'm also going to get this paper uploaded in today's video description along with the last two paper. But if somebody wants to attempt it live, the question paper is visible and we'll keep changing this page. So let's discuss this paper first, and then we will discuss some of the questions which were left in the last paper. So there are two things I will remember for this year prelims examination. One is NextCar 19, another is BIRSA 101. Let me write that also. So there is not a dedicated question on this theme, but I will prepare this also. The first CRISPR-based treatment option for sickle cell anemia that was approved in India just in month of January. And because of that, this topic is also very, very important. So know more about sickle cell anemia, know more about CRISPR gene editing itself. But in any case, ये जो topic है, this has to do with what is called CAR-T therapy. And that is a cancer treatment method. So I've not used the word CAR-T therapy, but the answer here is D. It is a novel cancer treatment that cost at a fraction of the price in India compared to many other countries. That is the correct answer. So let me briefly tell you about what CAR-T therapy is. In CAR-T therapy, how this will function here is that if somebody is suffering from some specific type of cancer and clearly body is not able to fight it off, body is not able to destroy that tumorous growth. So in this case, the immune cells, for example, one of the most important part of our immune system is T cells. So we isolate T cells from blood, and we modify genes of T cells, and we add what is called chimeric antigen receptor, which simply means that now these T cells are capable of destroying specific type of antigen. In this case, that antigen is that cancerous growth. So that T cell is converted into CAR T cell now, and that CAR T cell is multiplied into millions of copies, and now that is transfused back to the patient. So now आप ऐसे समझ लो कि खुद का जो आपका एक immune system है, with help of course this modification which happened, is going to fight off that cancer. Now compared to this, if you look at the other methods such as chemotherapy or radiation therapy, they have a lot of side effect, and you have to go many times. From time to time you have to go for that. So compared to that, जो CAR-T therapy है, it is considered a better alternative for certain type of cancer. Few things should also be very clear, that cancer word can mean so many different type of cancers in different type of stages, and CAR-T therapy has only been approved for very specific type of cancer. It does not work as of now for all type of cancer. And in India, the first such approval occurred for this treatment, developed by Tata Cancer Research Institute, and the name of that treatment was NextCar 19. So answer here is D. I will prepare more about CAR-T therapy, what it can do and what it cannot do. And I will prepare more about NextCar 19. ये more than enough है. NextCar के बारे में इतना जानना more than enough है. But I will also prepare CAR-T therapy as a topic in detail. This is a topic which has not been asked by UPSC. क्या होगा आंसर इसका? A. Epigenetic changes. So we often talk about mutations. Whenever we study about DNA, we study about mutations. Mutations are this irreversible spontaneous change in your base pair sequencing, right? So you inherit 50% of genes from your father, 50% from your mother. But in the course of life, due to errors in DNA replication, due to viral infection, due to certain chemicals, or certain type of radiation such as ultraviolet light, X-ray, gamma ray, this irreversible spontaneous change in DNA structure can happen, which is called mutation. That cannot be reversed back. But turns out there is another type of change, and we do not understand it a lot, by the way. And this change is called epigenetic changes. In epigenetic changes, think of it like this, that certain genes are being switched on and switched off. And why they are being switched on and switched off? we have some idea, but we do not understand it really well. So for example, even though your DNA base pair has not changed permanently, that has not happened, it's not mutation. For a brief duration, a particular gene is not functioning. A particular gene which should express itself is not expressing itself. Again, it expresses later. But for the time, if it does not express, which was required, you can suffer from diseases. What we know so far is that certain type of things that influences epigenetic changes, even among young people. So if you're not sleeping well, if your diet is not good, if you are not exercising, if you're not very active, so very basic thing, your mental health, by the way, your mental health can cause epigenetic changes. Your physical health can cause epigenetic changes. So this is an emerging branch of medicine that let's understand more about why does it happen. We have some idea, but we do not understand each and everything. And two of the best example of epigenetic changes, one is called DNA methylation, another is called histone modification. So in DNA methylation, a methyl group attaches itself to one of the base pair. Methyl group is CH3. In histone modification, one of the proteins which is your DNA is very nicely packaged inside that thread-like structure called chromosome, and that protein in itself undergoes certain changes. But it is not a permanent change. Right? That change for a brief duration can silence a gene. But again, that change can be reversed. So what I would expect you to do, that read more about epigenetic changes. I hope this is now very clear, this is our third session, that each and every topic we are discussing, it is not only the question, it is the theme in itself which should be studied in more detail. So read more about epigenetic changes and read more about DNA methylation and histone modification. No, RNA interference does cause gene silencing, but it is not exactly epigenetic changes. RNA interference is a different mechanism through which a particular gene can be silenced. So that's a different mechanism, we studied that separately, right? This happens because of certain type of chemical changes happening in DNA. And those chemical changes can be influenced by a lot of other factors. So your premise is true, that RNA interference does cause gene silencing, but it is not epigenetic changes. This is not a recent current affairs, ये 2 साल पुराना current affairs है अब तो, या 1.5 years पुराना current affairs है, but I still think we should know about this. क्या होगा आंसर यहाँ पे? Answer is, it regulates gene expression after transcription by binding to messenger RNA and either degrading it or blocking its translation into protein. Okay, so इसको आप ऐसे समझ सकते हैं. दो तरीका इसको समझने का, बहुत बार एंड आपका आरएनए इंटरफेरेंस वाला टॉपिक आ जाएगा इसमें. सो बहुत बार ऐसा होगा दैट एक तो सबसे सिंपल तरीका इसको समझने का ये है दैट इवन दो ऑल ऑफ आवर सेल्स हैव एग्जैक्टली सेम कॉपी ऑफ डीएनए, बट नॉट इन एवरी सेल सेम जीन इज एक्सप्रेसिंग इटसेल्फ. जैसे जो लिवर जब एक्सप्रेस, लिवर में जो प्रोटीन्स बनेंगे वो सेम प्रोटीन किडनी में नहीं बनेगा, वो सेम प्रोटीन हार्ट में नहीं बनेगा. तो प्रोटीन सिंथेसिस के लिए a part of DNA can reach to ribosome and the protein synthesis can start happening. I have one more question on this topic, by the way, on protein folding. But the fact that despite the very simple thing is that all cells have same copy of DNA, but not all genes are expressing in each and every cell. तो पहला तो एक ऐसे भी हम अप्रिशिएट कर सकते हैं कि there must be some mechanism which is dictating that कुछ genes एक organ में एक्सप्रेस होंगे, कुछ दूसरे जीन किसी दूसरे ऑर्गन में एक्सप्रेस होंगे. फर्स्ट. सेकेंड, टर्न्स आउट दैट समटाइम्स that some of the genetic expression may not be good. So gene expression is important for protein synthesis, but sometimes it may not be good for overall your health. And body has this internal mechanism which essentially can ensure that either gene does not express itself, or even if it does express, even if messenger RNA molecule is made, then that does not lead to actual protein synthesis. So ये जो collective mechanism है, this is called RNA interference, which you said, and that RNA interference in itself can happen with many ways. For example, there is something called double-stranded RNA molecule which can cause that. There is a silencing RNA molecule, small interference RNA molecule, which can cause that. And then you have this microRNA molecule which can cause that. So the Nobel Prize was given to these two scientists, that they were one to figure out the mechanism through which, even if transcription has occurred, even if DNA has transcribed into messenger RNA, but that messenger RNA molecule does not translate to protein synthesis. So this is often called post-transcriptional gene regulation. जो microRNA है, if I have to use a keyword associated with microRNA, it will be post-transcription gene regulation. Answer here is B. Another topic which has been in news a lot, and I do think that I should know about it, इसको AI से भी आप relate करोगे, जो पूरा artificial intelligence वाला हमारा theme है, तो आप उससे उसको relate करोगे. I told you in the previous class that एक तो LLMs के बारे में हम सबको पता है. We know about ChatGPT, we know about Claude, we know about Gemini. But apart from that, अगर आप Google में थोड़ा और Google के बारे में पढ़ोगे, तो Google का एक division है that is called DeepMind. And DeepMind is doing some really remarkable work apart from LLMs. So one of the tool they developed, this tool is called AlphaFold. And AlphaFold as a deep learning AI. So this deep learning AI was trained upon, थोड़ा सा background, let's discuss some background first. And I will come to this. देखो जो actual proteins हैं, they can be very, very complicated. The actual protein structure अगर आप देखोगे, they can be very complicated. But despite all of that complication, all proteins start with their building blocks called amino acids. Now there are many amino acids in the nature, we need 20 of them. So for protein synthesis, 20 amino acids are required. These amino acids, they come together, you can think of them as a pearl, and they are bonding together. This bond is called peptide bond, and they form a necklace-like structure. Imagine it like this. But that is not the final structure. Now it undergoes lot of processes, and the final structure will be this very complicated three-dimensional folded structure. So one of the open problem in biology was that if I give you this, if I give you this sequence of amino acid, can you tell me that final structure of that protein? And this problem was not solved by for many years, more than 20 years, this problem was not solved by any of the scientists anywhere in the world. And then in 2019, Google announced that we have this AI, which has been trained upon all of the known proteins and amino acid. And now if you give it a new sequence of amino acid, it will predict the final protein structure with very high accuracy. And they called this AI tool AlphaFold. Okay. So AlphaFold का अभी जो third version है, I've given link also. आप ये जाके थोड़ा पढ़ना इसके बारे में, ये जो deepmind.google है. And AlphaFold का जो third version है, it's so good that it has been able to predict final structure of protein for so many different proteins with such high level of accuracy that this was given Nobel Prize last year. So this is the question. AlphaFold develop. By the way, one more thing about amino acid, since I'm discussing it here, I hope you already know this. This is basic biology, that out of 20 amino acid, 11 can be made by our body automatically. But for that 9, we need to have some external food. So I need to have external protein source. And when you take that protein source, body first takes out amino acid of that protein, and then it makes its own protein. So those 9 amino acids which require external food source, these are called essential amino acid. So AlphaFold, developed by Google DeepMind, solved the protein folding problem with high accuracy, a fundamental problem that had stumped biologists for many years. This is true. Proteins are built from a standard set of 20 amino acids that combine in various sequences to form long chains. This is also true. What will be the answer, A or B? B. Why would answer be A? So answer will be B here. Both are individually true, right? But AlphaFold ने कैसे काम किया? How did it predict that? That is not getting explained by that second statement. अगर मैं थोड़ा सा ये बोलता कि AlphaFold was trained upon as a AI, as a deep learning tool, it was trained upon all of the known amino acid sequences and protein, and then how it learned to do that final prediction, तब answer A होता. So in this case, both statements are individually true, but it is not the correct explanation. Answer here will be B. Another topic which I think we should prepare, ये कुछ इसमें themes ऐसे हैं जो कि UPSC कभी ना कभी क्वेश्चन आएगा इस पे, वो इस साल आए अगले साल आए. Some of those themes are like that. So this is one of those themes. तो बहुत ही एक ambitious program कुछ साल पहले गवर्नमेंट ऑफ इंडिया ने लॉन्च किया. Within Ministry of Science and Technology there is a department called Department of Biotechnology. And they announced this project in 2019. The project was finally concluded in 2024. And all of that data right now is available on a supercomputer which is not very far away from here. Somewhere in Faridabad, there is a supercomputer. And all of that data is stored there. So this project was called Genome India project. The goal of the project was to gather gather DNA information of all the ethnic groups of India. And for the purpose of this research, and we already have done anthropological study about Indian ethnic population, and we figured out that there are about 4,600 ethnic groups in India. So from those 4,600 ethnic groups, we are going to collect 2 samples per group. So on an average, there will be 2 samples per group. So total sample which were collected were about some 10,000. और चूंकि हमको हर जगह थोड़ा सा एक रिलीजियस वो लेके आना होता है, so the actual sample was 10,008. So but anyways, but आपको ऐसे याद रखना पड़ेगा कि 10,000 samples were gathered, and the goal here was to do the complete genome sequencing and that becomes the reference genome of Indian population by ethnicity. It should be very clear that whatever data is being stored, it has been anonymized. Anonymized का मतलब ये किसका data है, that information is not available. ये किस group का data है, that information you can access, but ये किसका individual का data है, that will be a gross violation of privacy, that if we do not anonymize this data. There is a proper protocol you have to follow. If you are a researcher in India, चाहे public हो, private researcher हो, आप किसी Indian institute के साथ collaborate कर रहे हो, even if you are a foreign researcher, then there is a proper protocol you have to follow, and you have to apply for access to this data, and this data is going to be accessible to you. So public researchers के लिए, private institute researchers के लिए data accessible है, but ऐसा नहीं है कि freely available है. मतलब there is, ये depend करता है कि आपका purpose क्या है, उसके हिसाब से फिर what level of data will be accessible to you. All of that is part of one of our data sharing policy. So in any case, it aims to sequence the genome of large number of Indians from diverse ethnic backgrounds to create a reference database for Indian genetic diversity. This is true. Indigen. Now, Indigen was a project by CSIR. This was similar but not as ambitious as Genome India project. Indigen project का purpose जो था ये था that to figure out the genetic diversity within Indian population first, and to also make this gene testing mechanism cost effective so that it becomes accessible for Indians. जैसे मान लो मुझे अपना DNA test कराना है, that if I have some genetic predisposition to some disease or something like that, then it should be cost effective for me. So that was also one of the goal of Indigen project. So similar but not exactly same as Genome India project. So that sequenced genomes from over 1,000 Indians and found thousands of genetic variants unique to Indian population that were not in global databases. This is also true. This is one of the finding of Indigen project. Genome sequencing data of Indian citizen is currently open access and freely downloadable by any researchers. ये गलत है. Third जो है वो गलत है. ये कुछ ज़्यादा ही हमने इसको easily, we have data sharing policy, but जिस तरीके से ये description है ये गलत है. Answer here will be only 2. So in this theme, read about genome sequencing first, read about techniques of genome sequencing second. So for example, you should go and read about that when genome sequencing as a technique was developed, it was developed by a scientist named Sanger, that is called Sanger sequencing. And Sanger sequencing, however, was a very slow process, that it takes fragments of DNA and then it reads few of those fragments together. But the total genome sequencing as a result of that can take months and years also. We have 3 billion base pairs in human genome. So the second generation or next generation genome sequencing as it is called, that can do millions of fragments, it can read millions of fragments simultaneously. And that reduced that entire time of genome sequencing from few years or 1 year or few months to few weeks at max. And I should also tell you that there is a technique called nanopore sequencing, and that is often called third generation genome sequencing. And nanopore genome sequencing can do this entire thing within matter of few hours. So in nanopore genome sequencing, you start with nanopores, like literally you have holes of few nanometer diameter, and then as DNA molecules pass by through these pores, there are electrical signals acting as a biosensing mechanism to figure out that which of the base pair has passed through. So this technique is called nanopore sequencing. So not only I will prepare about Genome India project, I will also prepare about genome sequencing as a topic. I will prepare about what type of benefits genome sequencing can lead to. Jaise in India, it can allow us to understand more about the diseases, about genetic diseases first. It can allow us to understand more about genetic predisposition to certain diseases. It can allow us to know more about what is called personalized medicine. Personalized medicine का मतलब ये होता है that आपका जो treatment plan होगा that will cater to your specific genetic makeup. You belong to an ethnic group. Maybe there are certain medicines which will not work well for you compared to some other group. So, but अभी क्या है हमारा treatment option कि आपको ये disease है, ये medicine है, one size fit all approach. So, personalized medicine का मतलब ये होता है that आपका जो medicine treatment plan है, it is catered to your specific genetic makeup. So, it can help in that also, and it can of course help in understanding the evolutionary relationship or population genetics as it is called, that how different populations are same or different from each other, how one ethnic group might be related to another ethnic group, what type of migrations may have occurred, let's say a few hundred years ago, which caused a different group. So such type of anthropological study can be done with help of genome sequencing. So here answer will be only 2. This year, next year, 2 साल के लिए कम से कम ये टॉपिक में प्रिपेयर करूंगा. That is called GLP-1. क्या है आंसर इसका? Answer is A. They mimic the gut hormone, stimulating insulin release, slowing gastric emptying and reducing appetite. So that is how it works. एक आप ऐसे भी याद रख सकते हो कि this can have applications in diabetes treatment and obesity treatment and as well as even some benefit in cardiovascular diseases. So GLP-1 जो है बहुत काफ़ी फेमस ग्रुप ऑफ मॉलिक्यूल्स हैं. जैसे फॉर एग्जांपल ये होता है कि जब आपने देखा होगा कि आप बहुत हंग्री हो तो आपको कुछ भी अच्छा लग सकता है, बट जब आपने बहुत सारा खाना खा लिया है देन इवन इफ समथिंग वेरी डिलीशियस इज इन फ्रंट ऑफ यू मे नॉट लाइक इट, राइट. तो ये इसका मैकेनिज्म है, and once we understood that mechanism, that what type of molecules body produces which gives you that feeling of fullness, and then you would not want to eat things. So our body does that naturally. When scientists tried to do this artificially, they realized that जो body है इन molecules को बहुत immediately destroy कर देता है. So a lot of research happened for many years that how to ensure that ये जो molecules which we are providing, which mimics the behavior of naturally occurring molecules, I'm calling GLP-1, and how to stabilize that inside body. And they figured it out, that how do we do that. अब ये टॉपिक इसलिए न्यूज़ में था अभी that इसका जो पेटेंट है अभी एक्सपायर हुआ है, एंड इसका जो जैसे मान लो कि इसके अलग अलग डोज हैं, आप महीने में 4 आपको इंजेक्शन मान लो लेने हैं, जस्ट स्किन के बिलो आपको एक इंजेक्शन दिया जाएगा, 4 इंजेक्शन लेने हैं. So the total cost of those 4 injection came out to be some 9-10,000 rupees. The fact that now that has expired, that patent has expired, that means that now we can produce what is called generic version. So you'll find lot of names like Ozempic or Manuzaro. These are examples of this category of medicine called GLP-1, glucagon-like peptide. This is the full form. So ये Ozempic, Manuzaro जो मार्केट में, they are nothing but that GLP-1. So now इनका जो generic version है, this already is available in the market. अभी just 2 week पहले इसका patent expire हुआ था. The day patent expired, जितने भी top pharma companies हैं इंडिया के, they said that we have a generic version available, and now that cost is going to be about some 2-3,000 rupees from 10,000 rupees. So the goal here is that this medicine becomes more accessible. As a health tip, nobody here needs that. It is only and only for people who are suffering from serious obesity and serious diabetes as a problem, right? It's always a good idea to take care of healthcare by lifestyle changes rather than taking medicine. So in any case, the answer here is A. But ये जो mimic the gut hormone है, if once you are, जैसे मान लो आपका साल भर आपको डॉक्टर ने बोला कि साल भर आप यूज़ करो, एंड साल भर बाद जब आप वापस आओगे तो वापस से वो इंस्टिंक्ट ऑफ ईटिंग आएगा, तो इन एनी केस आपको अपना लाइफस्टाइल मैनेज करना ही पड़ेगा. वाई एम सेइंग दैट कि अगर स्टेटमेंट ऐसा आ जाए कि साल भर ट्रीटमेंट के बाद कोई ज़रूरत नहीं है, आप हमेशा ऐसा कुछ नहीं होगा. यू इन एनी केस हैव टू मेंटेन योर लाइफस्टाइल. तो आंसर हियर इज ए. So because of this entire concept of generic medicine also was in news. एक उस पे भी क्वेश्चन है generic medicine पे, तो वो भी एक टॉपिक आप प्रिपेयर करोगे इंडियन पेटेंट एक्ट के बारे में, the concept of generic medicine के बारे में. That as a topic also should be prepared. अच्छा, ये है ही, नेक्स्ट ही क्वेश्चन है. Not exactly about generic medicine, but more about the Indian Patent Act. Consider the following statements. India has specific legal provisions that protect patents' access to affordable medicine against strategies that pharmaceutical companies use to extend their drug monopolies. This is called evergreening of patent. That patent के लिए 3 criteria चाहिए. पहला criteria को हम बोलते हैं novelty. That whatever you are claiming should be something new. So this criteria is called novelty. The second criteria is called non-obviousness, which essentially means that whatever new you are claiming, it has to be inventive. Inventive का मतलब यहाँ पे ये है that it should not be very obvious change. कि जो भी आप new ये claim कर रहे हो, ये बहुत obvious नहीं होना चाहिए. एक minor सा आपने कुछ modification किया and you are saying that this is an inventive step. So if that's the case, then you do not get patent. And third is industrial use. That whatever invention it is, it should be have some application in industrial process or product. An abstract idea, that is not going to be patentable. So a lot of pharma companies, what they do, that 20 साल के लिए पेटेंट मिलता है आपको, पेटेंट का जो पीरियड होता है 20 साल होता है. Once that 20-year expires, generic versions will be available. So pharma companies will make some small minor claim and they will say that they have improved the efficacy of drug or something like that, and they will try to extend that patent period. So this strategy is called evergreening of patent, and in India there is a section called section 3D of Indian Patent Act which prohibits this practice. And in India this has been used few times in last 10 years to reject patent claim, to reject that renewal of patent claim. So first statement is true. Evergreening is a practice where a drug company files new patent, minor modification, that is also true. And third is also true, by the way, that compulsory licensing under India's Patent Act allows the government to authorize a domestic manufacturer to produce a patented drug without the patent holder's consent. But it can only happen under a very specific condition. देखो पहले तो licensing का मतलब क्या होता है, that should be clear. Licensing simply means that somebody is a patent holder. कोई कंपनी A है, उनके पास किसी प्रोडक्ट का पेटेंट है. I am कंपनी B, I go to them. I say that आप इतना ज़्यादा प्रोड्यूस कर नहीं पा रहे हो. Please share your technology with me. I am going to share my profit or I am going to pay some money to you. अब ये बिल्कुल पेटेंट होल्डर का प्रेरोगेटिव है that that patent holder gets to decide that if he or if that company wants to share patent or not. So if let's say they do decide, this is called licensing agreement. That licensing agreement will occur by paying some money from that company B to company A. मान लो there is a public health emergency, जैसे there is a global pandemic, epidemic going on. एक ये condition हो सकता है. And दूसरा condition एक और ये भी हो सकता है that a particular medicine is so expensive that in a developing country that will not be accessible to most of the people. So in these two cases, an exception can be created by government that even if the patent holder is not agreeing to license agreement, government will say some domestic company that you go and start making medicine, you do the reverse engineering, you start go and making medicine, we will take care of the legal issue. There is not going to be any legal issue. So this provision is called compulsory licensing, and ये खाली India specific नहीं है. This can be done by any developing nation in the world because it is part of a global agreement called TRIPS agreement. अभी WTO वापस से न्यूज़ में है, WTO राउंड ऑफ नेगोशिएशन चल रहा है, so आप वो भी प्रिपेयर कर लेना. But WTO का एक राउंड ऑफ नेगोशिएशन जो कंप्लीट हुआ था 90s में, इनफैक्ट इट लेड टू वन ऑफ दैट provision called TRIPS, Trade Related Aspects of Intellectual Property Rights. And that TRIPS allows compulsory licensing. And in India, there is a provision for compulsory licensing. So here answer is C, 1, 2 and 3. Okay. Question number 8. ये तीनों हम डिटेल में भी पढ़ेंगे हम. ये जो तीन टेक्नोलॉजी दे रखा है, by the way, what will be the answer here? Answer is 1, 2 and 3. All three are being used here as a technology. ये पहले तो I hope that everybody knows about this news. That there is a company called Colossal Biosciences. This is a biotech company and उनका एक गोल ये है that कुछ जो extinct species हैं across the world, let's try to resurrect them. And recently they made a claim that they have been able to resurrect dire wolf, a wolf species which lived in US some 9-10,000 years ago. By the way, वो claim fraud claim है. Fraud, sorry, I will not use the word fraud, in the sense that how it is being marketed, that they have been able to resurrect dire wolf. That part is not correct. What they have been able to do, so देखो इसमें किया क्या जाएगा, that आप इनका कोई फॉसिल मिला, 1000 साल पुराना फॉसिल है, बर्फ से मिला आपको, आइस में दबा हुआ है, and you are able to recover like some part of it. Now it should be very clear that you cannot get the entire DNA molecule in exactly preserved form. It will be damaged DNA. So what you will do, with help of CRISPR-Cas9 gene editing technique, you will try to reconstruct that DNA as much as possible. Then you are going to do that cloning technology, which is called somatic cell nuclear transfer technology, that you will take some egg cell of a relative species. That same species का मान लो अभी कोई relative species अभी जो exist करता है, जैसे मान लो woolly mammoth became extinct, but जो elephants हैं they are closely related to woolly mammoth. Dire wolf is extinct, but जो grey wolves हैं जो North America में अभी मिलते हैं, they are evolutionary relative of that dire wolf. So with help of cloning technology, we are going to use this technology called somatic cell nuclear transfer, and then we are going to put that in the womb of a mother. So for example, that woolly mammoth egg cell that is going to be placed in the womb of elephant. So the baby will essentially have some component, at least some physiological traits, which will be similar to what this species existed in the past. तो ये जो dire wolf वाला claim है that they have been able to bring back dire wolf from extinction, that claim is not entirely true. This is also a fact that lot of physiological traits, so those babies are visible. आप देख सकते हो जाके picture पे, अभी babies हैं, dire wolf babies they were able to have, and some of their physiological traits, it's very similar to what might have existed 9-10,000 years ago. And so that is backbreeding, by the way. Backbreeding means that you try to find evolutionary relative right now, which still is alive, and then with help of that relative, you're trying to again resurrect that particular species. So answer here is 1, 2 and 3, answer is D. But I will prepare about these three things, at least the first two, also in detail. I will prepare about CRISPR-Cas9 technique, I will prepare about that somatic cell nuclear transfer technique in more detail. And of course I will prepare about this entire concept of extinct species. Also try to know more about that where this species exists. जैसे तस्मानियन टाइगर ये कहाँ पे मिलता था? तस्मानिया, where is that? ऑस्ट्रेलिया के नीचे है, हाँ तो ये जरूरी है, right? तो ये पता होना चाहिए. Woolly mammoth in Arctic region, right? Dire wolf in North America. Okay, question number 9, what is the answer? Anyone attempted? D is the correct answer. The word exposome, पहले तो exposome as a word पता होना चाहिए. Why this question? बहुत साल पहले 1990 में एक प्रोजेक्ट था जिसको हम बोलते हैं ह्यूमन जीनोम प्रोजेक्ट. What Department of Biotechnology started in 2019, that Genome India project. उसी का एक जो सबसे पहला वर्जन जो दुनिया में हुआ था, that was called Human Genome Project. So Human Exposome Project. Exposome as a word means that from birth to death, all of the factors which you are exposed to, the physical, the chemical, the biological. For example, cities में air pollution है, diet आप क्या खा रहे हो, everything you are exposed to, how that impacts your health. So this is a very ambitious project which is going on right now, and you can easily appreciate that this as a project can be considered more difficult than Human Genome Project. बिकॉज सबसे पहले तो ये जो फिजिकल, केमिकल, बायोलॉजिकल कंडीशंस हैं ये दुनिया में डिफर करेंगे. अगर आप दिल्ली में रह रहे हो वर्सेस कोयम्बटूर में रह रहे हो तो आपका जो एक्सपोजोम कंडीशन है दैट इज डिफरेंट. दिल्ली में एयर पोल्यूशन का क्या इंपैक्ट होगा इन लॉन्गर रन, 10 साल आप दिल्ली में रह गए, यू विल हैव टू फिगर दैट आउट, राइट. सो एक्सपोजोम प्रोजेक्ट का गोल ये है दैट लेट्स select certain places, different geographical places, then let's select people living in those places and let's target few people across many years and then see that how those physical, chemical, biological factor impact their health. So this is a very ambitious project going on right now. This project has not been completed. This will take many years to be completed. So this project is called Human Exposome Project. Answer here is D, the totality of environmental exposure, chemical, physical, biological and social a person experiences from conception to death. So know about the project and know also about the meaning of that word exposome. That what do I mean by that word? Exposome. Next question. One Health. As a thumb rule, remember that even if let's say there is not a question in prelims examination, use this as a keyword in your mains answer writing. That अगर health related कोई भी question आता भी है या environment versus health related कोई question आता है, ऐसे लिखना हो आपको, write about One Health. There is a growing emphasis, by the way, what is the answer here? There is a growing emphasis on One Health approach that recognizes that human health, animal health and ecosystem health are interconnected. This is a fact. One Health का literal meaning ये है that human health cannot be thought of in isolation, right? Second statement is that many new infectious diseases are zoonotic in origin, that is they originate in animals and spilled onto human population. Third is that deforestation and wildlife trade, which increases contact between humans and wild animal reservoirs, are recognized risk factor for pandemic emergence. So all three are true and statement 2 and 3 are correct explanation of statement 1. Answer here is A. So in this question, that why we have to think about that so this is what I meant by human exposome. In any case, how should we think about One Health approach? That you should not think about human health in isolation. That in the environment, in the surrounding you live, think of the animals you come in contact with. So we have to do some sort of early warning system even for animals, or think about the environment you live in. So how pollution impacts your health, how lack of greenery impacts your health, how sound pollution impacts your health, the type of food you eat, जहाँ पे pesticides ज़्यादा है, how does that impact your health? So this study, this integrated approach of health, where we have to think about these three things in conjunction, is called One Health. So all are true and answer is A here. Why did you think B? कौन सा statement गलत लगा था आपको? Third statement you even though thought to be correct but is not a explanation, right? But the moment this is true, then third should be true and should be explanation. पहले तो ऐसे 3 स्टेटमेंट का देखो, एक अच्छी चीज़ ये है जिस तरीके से यूपीएससी ने फ्रेम करा है 3 स्टेटमेंट वाले क्वेश्चन को, दैट यू डू नॉट हैव टू वरी अबाउट फर्स्ट स्टेटमेंट अनलेस स्टेटेड अदरवाइज. जैसे मान लो कि यूपीएससी ने थोड़ा सा फिर से अपना पैटर्न चेंज कर दिया 2026 में, एंड उस पैटर्न में ऐसा हो गया है कि दे आर नाउ रेजिंग अ डाउट ओवर इवन स्टेटमेंट 1. जैसे जो स्टेटमेंट 2 वाले स्टेटमेंट में क्वेश्चन होता है कि ईदर ऑफ द स्टेटमेंट कैन बी रॉन्ग ऑल्सो. जो 3 स्टेटमेंट वाले क्वेश्चन जो इन्होंने लास्ट टू लास्ट ईयर इंट्रोड्यूस किया, लास्ट ईयर कम से कम 8 क्वेश्चन थे इस पे. सो इसमें एक जो लास्ट ईयर इन्होंने किया था कि स्टेटमेंट 1 इज ट्रू, यू हैव टू थिंक अबाउट नाउ स्टेटमेंट 2 and 3. So you have to be also prepare for a scenario that maybe even there is a doubt on statement 1. So if that is stated in the option, then you have to think about statement 1. And if it is not stated in the option, if the option is like that, then I know that statement 1 is correct. I only have to worry about statement 2 and 3, that are they correct? And if they are correct, are the explanation of statement 1. So here the answer is A. Question number 11. Generic medicine is chemically identical to original branded drug whose patent has expired. That is a fact, that's true. Patent expires after 20 years. नाउ यू कैन मेक जेनेरिक मेडिसिन. आप बस ब्रांड नहीं बेच सकते, आपको उसके एक जेनेरिक नाम से बेचना पड़ेगा. आप खुद का अपना नया ब्रांड नहीं बना सकते ऑफ जेनेरिक मेडिसिन. बट फर्स्ट स्टेटमेंट इज ट्रू. Biosimilar is not an exact copy of the original biological medicine because biologics are large complex molecule. This is also true. That biosimilars essentially mean that we are talking about complex molecules such as insulin मान लो complex molecule है. बहुत सारी चीज़ें हैं जो complex हैं, vaccines हैं वो complex हैं. So biosimilar का मतलब ये है that wherever you have complex biological molecule, ये हम नहीं बोल सकते कि दो चीज़ें exactly identical हैं. There might be a small difference, but that's fine. Even if that small difference is there, जो purpose biosimilar का है, जो purpose medicine का है, that it should have some therapeutic effect. So it still will have that same therapeutic effect, even though it is not an exact 100% identical copy. So biosimilar is a word used for again those medicines which are biological in nature and whose patent has expired. Right? So a generic version can be made, but that generic version will not be identical copy of the actual medicine. But even then, it will have that same therapeutic effect. So answer here is both 1 and 2. तो अभी बायोफार्मा शक्ति जो स्कीम गवर्नमेंट ने बजट में लॉन्च किया था, उस पर भी एक क्वेश्चन है. उसमें आप जाओ तो एक वर्ड है कि बायोलॉजिक्स को लेके हमको इन्वेस्ट करना है. सो बायोलॉजिक्स एज आई सेड आर कॉम्प्लेक्स मॉलिक्यूल्स कंपेयर टू सिंपलर मॉलिक्यूल्स जो आप यूज़ करते हो, जैसे पैरासिटामोल हो गया, एक सिंपल सा मॉलिक्यूल है, उसका एक इम्पैक्ट है. बट जो बायोलॉजिक्स हैं, दीज आर कॉम्प्लेक्स मॉलिक्यूल्स, बायोलॉजिकल मॉलिक्यूल्स विथ सम थेराप्यूटिक इफेक्ट. सो एक जो फोकस है अभी, दैट इफ यू वांट टू हैव न्यू मेडिसिन, द न्यू मेडिसिन में हमेशा एक प्रॉब्लम था व्हेन द क्लिनिकल ट्रायल्स वर डन in 1960s and 70s, that you isolate some cell and tissue and then you do testing upon it in a lab petri dish. There was always a concern that it does not mimic the actual behavior of that same cell and tissue when it is part of a large complex system. So आप ऐसे समझ लो कि some liver tissue, how it will behave in a lab petri dish versus how it will behave in a complex setup around liver are two different things. So in this context, lot of advancements have occurred in pharma sector, that how can we make our clinical trial, drug trial more efficient. And यहाँ पे ये जो दो चीज़ें हैं इनके बारे में पता होना चाहिए. That organoids, these are miniature simplified version of human organs grown in a lab from human stem cell, that is true. And जो organ on a chip है, that is even better. That not only it is that tissue, roughly the size of USB drive, but if a particular tissue, जैसे मान लो जो हमारे lung tissues हैं, and what type of physical, chemical, mechanical changes they undergo through, because they are part of a system, you're trying to replicate the same scenario on that chip, that you can control that physical factor, that chemical factor, that mechanical factor, and then you try to see that how that specific tissue is behaving. So you're trying to mimic the system itself. It's not just the tissue. You're trying to create a small system, a miniaturized system, that how that tissue will behave inside human body, and that is called organ on a chip technology. And that three-dimensional 3D bioprinting is also true, that using a modified 3D printer that uses living cells to print three-dimensional tissue structure layer by layer. Answer here is all of the above. Okay. C is the correct answer. You can think of organoids like this, organ on a chip, and 3D bioprinting. एक कॉन्सेप्ट होता है, चलो ठीक है आप पूछ, एक 4D प्रिंटिंग का एक कॉन्सेप्ट है. तो 4D प्रिंटिंग जो है अगेन एक बढ़िया मार्केटिंग वर्ड है. यहाँ पे जो 4 डायमेंशन उसको बोल देंगे कि टाइम इज द 4th डायमेंशन. बट एक्चुअल व्हाट इट इज दैट आप जो 3D प्रिंट करके प्रोडक्ट आपका 3D प्रिंट होके आया, द टाइप ऑफ मटेरियल्स यू यूज्ड इन दैट 3D प्रिंटर, दोज मटेरियल्स स्टिल कैन टेक सम अदर फाइनल शेप डिपेंडिंग अपॉन इन्वायरमेंटल एक्सपोजर. कि मान लो आपने उनको पानी में रख दिया या किसी ऐसे कंडीशन में रख दिया जहाँ पे pH हाई है या लो है, और मे बी यू एक्सपोज देम टू सम सॉर्ट ऑफ मॉइस्चर, and based on that they are taking their final shape. So the final shape, the final design is not the actual printed design. The final design is the environmental exposure in time, and hence the name 4D printing. And yes, you are right. So description है वो सही है. That 4D printing simply means कि जो आपका printed product है, that still can undergo some final modification depending upon environmental factor. यहाँ पे क्या होगा आंसर? This is a question which you should attempt. Answer is C. ये clinical trial पे आप expedite नहीं कर सकते. Because think about it. Clinical trial we are doing to realize the safety of a medicine. That is a medicine safe or not. तो यहाँ पे अगर bureaucratic red tape है to remove the bureaucracy, that's a good thing. That bureaucratic red tape is not always a bad thing. So we are talking about human health here. As a result of that, ये ठीक है. अगर आप बजट में जाके देखो, बजट में अनाउंस किया था गवर्नमेंट ऑफ इंडिया ने स्कीम, 3 new institutes and जो existing institutes हैं, that has to be upgraded and 3 new has to be set up called National Institute of Pharmaceutical Education and Research. Enhance India's competitiveness in global biologic supply chain makes sense. हर statement को judge करने का ऐसे question में तरीका ये होता है that अगर ये true नहीं है तो इसका कौन सा part true नहीं है, I told you that, and इसका reverse फिर जो होगा वो कौन सा true होगा. जैसे मान लो to enhance India's competitiveness in global biologic supply chain. तो अब 2 कारण से एक false हो सकता है. एक तो ये है कि ये है ही नहीं स्कोप का, बायोफार्मा शक्ति में इसका स्कोप ही नहीं है, ये मेड अप स्टेटमेंट है. बट दिस विल नॉट बी ट्रू दैट इसका ऑपोजिट इज करेक्ट, दैट टू नॉट एनहांस इंडिया स्कोप. दैट डज नॉट मेक एनी सेंस. सो इफ इट इज देयर देन इट शुड बी ट्रू. अब ये दूसरी बात है कि इट इज देयर और नॉट, उसके लिए आपको फिर स्कीम पढ़ना पड़ेगा. दैट व्हाट आर द ऑब्जेक्टिव्स ऑफ स्कीम. डी, टू स्ट्रेंथन द रेगुलेटरी फ्रेमवर्क ऑफ CDSCO. This is also true that government ने specifically announce किया है that we need more trained workforce, more expertise in ensuring that हमारा जो regulatory mechanism है that matches with global regulatory mechanism. But see, that cannot be one of the objective to expedite clinical trial. What does that mean? Are you compromising upon safety of the medicine? So answer here is C. That is not correct here. Okay. Yes. But it was not expediting. That was, so you're right. Let me show you. I should have. You're right that there is. आप इसकी बात कर रहे हो ना? ये है. Creation of large-scale clinical research ecosystem with a focus on improving India's capacity to conduct clinical trial. Conduct is not same as expediting. So that is why that statement is wrong. So yes, there is a focus on clinical trial. बाकी सब है. Domestic development and manufacturing of high-value biopharmaceutical product. Expansion and strengthening of these institutes, National Institute of Pharma Education, NIPR. CDSCO to be reinforced to strengthen regulatory framework. बाकी सारी चीजें हैं, स्कीम का पार्ट है. This was in news. This is not a recent discovery. This was discovered in 1980s. That is called jumping genes. That part of your DNA, which are called transposons, that they can cut or copy themselves into some other part of DNA. And when they do that, they can change the genetic expression. So this mechanism is called jumping gene mechanism. Answer here is B. And this was in news recently, why this topic has been there. So remember this keyword also, transposons. Remember this part also. Sorry? It's not mutation. Jumping gene is not same as mutation. It can, that is a separate part. But mutation, when we think of the mutation, the word here is that some irreversible change in base pair sequencing. Okay. ये जो क्वेश्चन है, what is the correct answer? Sorry? No gene drive has yet been approved for environmental release anywhere in the world. That is actually true. A is the correct answer. And the reason it has not been approved because B is also true and also the correct explanation. ये टेक्नोलॉजी हम कर सकते हैं. लेट मी टेल यू अबाउट जीन ड्राइव टेक्नोलॉजी. The idea of gene drive technology is that CRISPR-Cas9 gene editing is so powerful that generally, let's say a particular gene which is responsible for specific trait, generally there is a 50% probability that it will go on to the next generation, right? So if I go 3-4 generation among large population chunk, few people, few of these species will exhibit that particular genetic trait, right? जैसे मान लो कि blue eye is a genetic trait. Let me give you a random data that blue eye is a genetic trait, right? So across the particular gene is responsible for that, and there is a 50% probability that it goes on to the next generation. Gene drive technology का मतलब ये है that with help of CRISPR-Cas9 technique you can ensure that a specific genetic trait is inherited by almost every baby. So more than 90% of the babies will have that specific genetic trait. So this technology is called gene drive technology. That rather than nature taking its course, you are doing gene modification in a way that a specific genetic trait is inherited by almost all of the babies, and their babies, almost all of them. So the goal of gene drive technology is that जो typical Mendelian law आपने पढ़ा होगा, that there is a 50% probability of a particular trait to be inherited, you are making that almost 100%. or definitely more than 90%. But more than 90% probability का मतलब ये है कि 3-4 जनरेशन बाद वो जो जेनेटिक ट्रेट होगा वो सब में होगा. इसको एक तरीका जो सोचा गया था, that let's use gene drive technology to cause a particular genetic trait where mosquitoes become essentially sterile, that they cannot produce babies. And as that genetic trait spreads, then essentially what you're trying to do here is that in a local area, the entire mosquito population comes to a collapse. So this is generally इस type का अगर extreme statement हो, that with help of gene drive technology, a particular species population can be collapsed. तो generally इस type का extreme statement आप गलत करोगे, but इस case में सही होगा ये. The gene drive technology can cause that. But even though mosquitoes are disease carrier and they can spread a lot of diseases, that does not mean that they do not have ecosystem functions. Right? They do play important ecosystem function. And yes, some of the mosquitoes, they cause some of the most prevalent diseases we know, but that does not mean that population collapse is the way forward. So that is why even though theoretically and in a small experimental we know that this is possible, this has actually not been done on a larger scale anywhere in the world. And the answer is that why it has not been done. So that statement 2 is also true, that once released into wild population could spread through an entire species within few generations and cannot be recalled. The reason why it can spread, because it allows a particular trait to be inherited with more than 90% probability compared to the typical natural law of 50%. So answer here is A, both are correct, and statement 2 explains statement 1. नहीं, वोल्बाचिया जो है वो दो तरीके से किया जा सकता है. वोल्बाचिया जो है आप उसको पॉपुलेशन कंट्रोल करने के लिए कर सकते हो, राइट. सो एक पॉपुलेशन कोलैप्स भी किया जा सकता है, बट यू कैन कंट्रोल द पॉपुलेशन. सो व्हेनेवर वी टॉक अबाउट वोल्बाचिया के साथ अच्छी चीज़ ये है दैट डिपेंडिंग अपॉन हाउ यू यूज इट, you can cause control or you can cause collapse. So here it can cause collapse, and because of that there is a fear. In Wolbachia, if you want to do, you'd ideally go for the control of the population, and that should be the way forward. That let's not focus upon the collapse of the population, let's focus upon controlling the population. Approved in the sense that a particular country has given go away that let's implement this. No, the answer is no. No country has done it. Some last questions. The reason I have added these questions here is that अगर आप पिछले 3-4 साल में देखो तो पहले से भी आता था, but पिछले 3-4 साल में इस पे और ज़्यादा focus है. That when we talk about some of the environmental problems we face, and then UPSC is expecting you to know about some of the technological solution to solve those problems. जैसे हम मान लो climate change की बात करते हैं, so you should know about solar geoengineering techniques, or you should know about carbon capture utilization storage techniques. When we talk about air pollution, water pollution, plastic pollution, then you should know about that what type of technological solution are being proposed for that. So that is a larger theme. So apart from the questions I'm going to show, try to read more about this theme also, because it typically environment का topic है, but ये environment science and tech का जो एक cross section होगा वहाँ पे ये fit बैठेगा. So whatever themes we have discussed till now, चाहे वो space हो, computing हो, biotech हो, apart from all of that, add one more theme in that technology part, that how different technologies can help in solving environmental problem. So I have only discussed few of them, but you go and prepare more of them in next few days, right? So जैसे एक सिंपल सा तरीका ये होगा that आप एयर पोल्यूशन से स्टार्ट करोगे, that आप पहले तो फोकस करोगे कि एयर पोल्यूशन जब हम बात करते हैं which are the important pollutants. So you will read about that, ठीक है, particulate matters are important pollutants, we should think about them a lot, because whenever we think of air pollution, सबसे पहला हम बात करेंगे PM 2.5 and PM 10 के बारे में. Then you will talk about, okay, what to do about volatile organic compounds, what to do about ammonia, what to do about oxides of sulfur, sulfur dioxide, what to do about that nitrogen oxide and nitrogen dioxide, what to do about ozone. So we'll ask about all of these questions. And then you will try to find out the specific solution to tackle those pollutants. So जो electrostatic precipitator है, it removes particulate matter, that is true, from industrial flue gas by giving particles an electrical charge and then attracting them to oppositely charged collecting plate. नाम से भी देखो कुछ तो लग रहा होगा, that we are using electricity to precipitate a particular pollutant. But second में नाम से कुछ भी नहीं लगेगा यहाँ पे. So second जो है, it is not carbon dioxide, it is NOx, it is NO or NO2. जो selective catalytic reduction technology का जो focus है, that is on nitrogen oxide and nitrogen dioxide. And every word has a meaning here. Selective का मतलब ये है that मान लो आपका vehicle है, vehicle emission में बहुत सारी चीज़ें release होंगी, hydrocarbons होंगे, थोड़ा बहुत sulfur dioxide होगा, particulate matters होंगे. So selective का मतलब है that we are selectively reducing का यहाँ पे ये मतलब है that NO and NO2 has oxygen, I am removing that oxygen. So that becomes a reduction reaction. And this reaction requires a catalyst, hence the word catalytic. And that catalyst is essentially ammonia. In fact, I can show you the reaction. You do not have to memorize it. This is the reaction. 2NO plus 2NH3, 2N2 plus 3H2O. That in the engine, NO or NO2 is being produced, but in the exhaust gas, a larger part of that NO and NO2, that is becoming nitrogen. And nitrogen is not a pollutant. So this technology is called selective catalytic reduction. We can also do non-catalytic reduction. Non-catalytic में क्या होगा कि हमको catalyst की ज़रूरत नहीं पड़ेगी. The only problem is that that will require very high temperature reaction. But in vehicles, for example, जो भी BS6 vehicles हैं, Bharat Stage 6 vehicles हैं, they are equipped with this technology of selective catalytic reduction. Answer here is one only. Okay, these two techniques, they belong to what is called bioremediation. In bioremediation, I talked about this in early classes also. In bioremediation, your focus is that you use microbial organisms, and then with help of those microbial organisms, you try to degrade pollutants. So अब देखो word जो है बहुत बार English word is going to help you. So if you are thinking कि इसको रटते हैं biostimulation and bioaugmentation, but even that if you know the meaning of the word stimulation and augmentation, that can also help you here. So biostimulation uses chemical fertilizers to kill contaminant microbes. Bioaugmentation uses natural predators. Biostimulation is used only for contaminated area. Only वाले word ऐसे भी आप थोड़ा सा इसको लेके आप ignore कर सकते हो. Biostimulation physically removes contaminated soil and treats it in controlled bioreactor, while bioaugmentation treats the contaminated in place without excavating the soil. That is in situ and ex situ. Biostimulation enhances the activity of naturally present microbes by adding nutrients, oxygen. Bioaugmentation introduces additional specialized microbes into the contaminated site to supplement native population. That is the correct answer. So stimulation का मतलब ये है that some microbes are already there, but even microbes require specific set of conditions to grow and prosper. and we provide those specific set of conditions. So you try to control the pH, you provide oxygen there, you provide moisture there. So this is called biostimulation. Augmentation का मतलब ये है that apart from microbes which are there, you try to add other microbes at that same place which can degrade pollutants even faster. So answer here is D. In general, ये जो पूरा concept of biofiltration या bioremediation है, ये बहुत important concept है, and that should be prepared separately for examination. I should have a slide on it. Yeah. That this entire concept of bioremediation and different strategies of bioremediation or biofiltration, it's a very important concept and should be prepared separately as a theme for prelims or even for mains examination, by the way. This is a question about that carbon capture, utilization and storage. That ocean alkalinization process enhances ocean capacity to absorb and sequester atmospheric CO2. By adding substances like lime or olivine, pH of ocean is increased, which promotes the formation of bicarbonate ion. What do you think is the answer? Answer is A. That if first is true, then second is also true, and second is also the correct explanation of statement 1. So there are many technologies together which are called carbon capture, utilization and storage. One of the problem you learn about as a consequence of climate change is acidification of ocean. That ocean is becoming more acidic. And one of the consequences of acidification of ocean is that marine organisms require an external exoskeleton, and that is called shell. And that shell is made of calcium carbonate. So that shell formation reduces because of pH value which is decreasing. But that shell formation is very useful because देखो shell जो है वो calcium carbonate है. Calcium carbonate at room temperature and pressure is solid. Many of those organisms, when they die, they sink to the bottom of the ocean. And for thousands of years, they're going to remain buried into that ocean. So you have CO2 captured from atmosphere, and that is being buried into the ocean. It's a natural process. But if ocean does become acidic, then this shell formation is affected, and that storage of carbon deep inside ocean, that is affected. So हमारा गोल यहाँ पे क्या होगा कि हम artificially हम alkaline materials such as lime, such as olivine, इसको हम add करेंगे, that even though pH is decreasing, you artificially increase pH so that that shell formation happens at higher rate, and more species, when they die, they bury deep into ocean. So more shell formation, more carbon capture and storage. तो देखो this is a natural process. इसी natural process को हम escalate कर रहे हैं with help of spraying some alkaline material on the ocean. बहुत सारे और तरीके हैं. There is something called ocean fertilization. In ocean fertilization, I should have a slide on that also. Okay, I do not have a slide, but let me explain ocean fertilization. In ocean fertilization, you spray certain things which can which can help in growth of phytoplanktons. Phytoplanktons are these microscopic algae which can do photosynthesis. So you are increasing the rate of photosynthesis done by marine organisms. So this is called ocean fertilization. कि आप fertilizer डाल रहे हो algal growth के लिए. आप इस तरीके से सोच सकते हो. So there are many technologies which enhanced rock weathering पे UPSC ने क्वेश्चन पूछ रखा है. तो ये तो मेरे हिसाब से नहीं आएगा जो enhanced weathering of rock है. That weathering of rock also captures CO2 and stores that CO2 in the soil. So इस पे तो UPSC क्वेश्चन 2 बार कम से कम पूछ चुकी है. But prepare this also. That what do I mean by this enhanced weathering process and how does that help into carbon capture and storage? So you have that series of technology called carbon capture and storage. Try to read more about them, and one of them I'm very sure that can be asked by UPSC in prelims examination. And they have made this trend of asking questions from environmental technology almost every year now. This is a question on water pollution control technology. Reverse osmosis, uses high pressure to force water through a semi-permeable membrane. That is true. So that salt is left and water passes by. This is opposite to a natural process called osmosis. So osmosis के लिए हमको कुछ भी external करने की जरूरत नहीं है, वो खुद से होगा. But reverse osmosis के लिए हमको energy supply करना पड़ेगा, and that is called RO. Coagulation का देखो हमेशा मतलब ये होता है that things ideally are so small that they remain suspended in the water. So in coagulation, your goal here is that they become some sort of, let's say, larger particles, and then with help of what is called sedimentation, that because of gravitational force, they accumulate at the bottom, and that filtering that pollutant, that large pollutant becomes easy. So electrocoagulation simply means using electric current to release metal ions, and these metal ions can react with those suspended solid particles, and that can now sediment, and that with now we can remove those pollutants from the bottom of the water. And UV, that causes ultraviolet radiation, and ultraviolet ionizing radiation जो है, that can cause damage to DNA of lot of microbial organism. इसका purpose है to kill microbe. So answer here is 1, 2 and 3. D is the correct answer. But there are again many more technologies related to water pollution. जैसे read about membrane bioreactor जो UPSC ने पूछा था. Read about coagulation and flocculation together. Read about zero liquid discharge technology. Read about activated carbon filter जो again last year अब पूछ रखा है तो इस साल तो नहीं आएगा, but फिर भी go and read about that what do I mean by activated carbon filter. Another topic of that carbon capture storage. In pre-combustion capture, fossil fuels are converted into a gas mixture containing hydrogen and carbon monoxide before combustion. In post-combustion, CO2 emission from flue gas is captured after fossil fuel combustion with absorbents. In oxy-fuel combustion, fossil fuel is burned in oxygen instead of air, resulting in the flue gas that are mainly water vapor and CO2. All three are correct here. ये जो है, this is nothing but coal gasification. Remember I told you about alternative energy में हम बात कर रहे थे, hydrogen production के अलग-अलग तरीके हैं. So one of that method I told you about is coal gasification. And I told you that coal gasification का एक सबसे बड़ा challenge क्या था? That lot of CO2 will be released. So in pre-combustion capture, आपका जो fuel है वो finally hydrogen है. You are burning that synthetic gas. Hydrogen is the fuel. CO2 is releasing before that. And then you capture that CO2 and you store it. So that is called pre-combustion capture. And if you remember, what color we assigned to this hydrogen? Blue hydrogen. We called it blue hydrogen. That even though CO2 is being released, we are capturing that CO2, not allowing that to be released into atmosphere. In post-combustion capture, you burn coal, let's say, and burning of coal will produce lot of gases together from those chimneys. So that mixture of gas is called flue gas, and now you have to filter carbon dioxide from that flue gas, and that carbon dioxide has to be stored somewhere in some geological repository, or maybe that carbon dioxide can be used into some industrial process. CO2 does have a lot of industrial application. तो यहाँ पे देखो एक बहुत बड़ा challenge ये होगा कि जो flue gases हैं, they will include lot of things, right? You have particulate matter, sulfur dioxide, oxides of nitrogen. उन सब gases में आपको CO2 को filter करना है, and that can become a challenge. So this is why this technology is called oxy-fuel combustion. That आप burning air में नहीं कराओगे, because air has lot of gases. The most important is nitrogen. आप कोल को burn करोगे air के presence में, तो nitrogen oxidize होगा, NO2 produce होगा. So you are not allowing burning to happen in air. आप पहले air को filter करोगे, and oxygen burning के लिए, coal burning के लिए I only need oxygen. So this burning is now happening in the presence of oxygen. And in presence of oxygen, when this burning happens, that flue gas mostly will include water vapor and CO2. You can condense water vapor, remove water, and the gas which is now left here is CO2. तो आपका जो post combustion में जो CO2 filtration वाला problem था, you have solved that problem. But then you have another problem that you first have to filter air, and you have to have that combustion only and happening with help of oxygen. So here all three of them are true. Answer here is D, 1, 2 and 3. एक और टेक्नोलॉजी है जिस पे अभी तक कोई क्वेश्चन नहीं आया है, दैट इज कॉल्ड केमिकल लूपिंग कंबशन. यहाँ पे ऑक्सीजन फिल्टर करने के बजाय हम क्या करते हैं, दैट हम एक मेटालिक ऑक्साइड यूज करते हैं. सो वी बर्न कोल, एंड बर्निंग कोल के लिए जो ऑक्सीजन चाहिए, वी आर नॉट डूइंग दैट फ्रॉम एयर. वी आर यूजिंग अ मेटालिक ऑक्सीजन फॉर द कंबशन. दैट मेटल जो है दैट रिड्यूसेस, दैट मेटालिक ऑक्सीडाइज रिड्यूसेस, ऑक्सीजन is used for the combustion, but then this entire process is recycled and we again produce that same metallic oxide, and we can keep using that metallic oxide again and again. So hence the name chemical looping combustion. So chemical looping combustion also has this benefit that it allows combustion of coal in presence of oxygen. As a result of that, जो आपका flue gas है, that mostly will be CO2 and H2O. But rather than filtering oxygen from air, you are using some sort of metallic oxide for this combustion, and you are recycling that metallic oxide again and again, hence that name looping combustion, chemical looping combustion. So I'll prepare this also as a topic in one line, that how does it work. HEPA, यहाँ पे क्या होगा आंसर? A. आंसर इज A. जो HEPA फिल्टर है बहुत बार न्यूज में रहता है, high efficiency particulate air filter. So that HEPA filter is series of filter. The final filter का जो membrane है, it can filter out as small as some 0.3 micron particle, and that will include a lot of pollens and certain type of bacteria also. It will not be able to filter out gases. Gases are much more smaller. It also cannot filter out solid particles of nanometer dimension, जो 1 nanometer 10 nanometer का, these things cannot be filtered out. But 0.3 micrometer, that also is very fine pollutant. Lot of pollen, certain bacteria, they can be filtered out with help of HEPA. So answer here is A. If you have question in any of these questions, like something you would want to add, would want to ask. There are few more questions. हाँ, sorry, go ahead. You have to be कौन से question में? Question number 11. हाँ जी, थोड़ा सा लाउडर होना पड़ेगा, कम आवाज आ रहा है, फिर से बोलिएगा. मतलब मैंडेटरी का मतलब? ओके, तो आप बोल रहे हो कि एक जो दूसरा तरीका है वो हो सकता है जेनेरिक का, वो हो सकता है कंपल्सरी लाइसेंसिंग वाला. दो ही तरीका है जेनेरिक बनाने का. जेनेरिक मेडिसिन दो ही तरीके से बन सकता है. एक तो यह है पेटेंट एक्सपायर हो गया है, एंड दूसरा जो मैंने आपको बताया कंपल्सरी लाइसेंसिंग, कि पेटेंट एक्सपायर नहीं हुआ है बट दैट मेडिसिन इज सो एक्सपेंसिव दैट पीपल कैन नॉट बाई इट. इन दैट केस गवर्नमेंट कैन आस्क सम डोमेस्टिक मैन्युफैक्चरर कि आप जाके इसका जेनेरिक वर्जन बनाओ. तो इवन दो पेटेंट हैज नॉट एक्सपायर्ड, जेनेरिक बनाया जा सकता है. मैंडेटरी नहीं है, बट ऐसा लिखा भी नहीं है कि ओनली हुज पेटेंट हैज एक्सपायर्ड. सो दिस स्टेटमेंट इन इटसेल्फ इज नॉट रॉन्ग, दैट जेनेरिक वर्जन विल बी अ मेडिसिन जिसका पेटेंट एक्सपायर हो गया. तो ये स्टेटमेंट खुद में सही है, बट आपका पॉइंट सही है. तो देयर इज वन मोर वे हाउ वी कैन मेक जेनेरिक मेडिसिन इवन इफ पेटेंट हैज नॉट एक्सपायर्ड, एंड दैट इज कंपल्सरी लाइसेंसिंग, अ टॉपिक आई एक्सप्लेन्ड फ्यू मिनट्स अगो. ठीक है, एनी अदर क्वेश्चन? यस. नहीं, तो आप ऐसे क्यों मान रहे हो कि 50%, सॉरी 50% अगर है? नहीं, तो 50 का 50 तो 25 होता है ना. 50% का 50% तो 25% होगा ना. सो दैट इज वाई आप ऐसे देखोगे कि मान लो एक स्पेसिफिक जीन है, जैसे मान लो कि पेरेंट्स में कोई एक पेरेंट के पास एक स्पेसिफिक जीन है, एंड इफ यू इनहेरिट दैट जीन you have that specific trait. You have some babies, if your babies inherit your gene, they have that specific trait. But they can also inherit gene from the mother, and in that case, maybe they do not have that trait. So that is why when you look at the natural occurrence, you find that a specific gene का जो inheritance pattern है, वो रहेगा, ऐसा नहीं क्यों खत्म हो जाएगा. अगर वो खत्म तब होगा अगर उस दैट इज कॉजिंग सम डिसएडवांटेज इन योर सर्वाइवल रिप्रोडक्शन, दैट्स अ इवोल्यूशनरी पार्ट. बट इफ देर इज अ ट्रेट which is not as such increasing or decreasing your chance of survival, then there is always a 50% chance that it will go on to the next generation. But in gene drive technology, you want that to be much, much more than 90%. A few more questions which we did not discuss yesterday. Any other question, by the way, in this paper? Yeah, we can make nuclear weapons from plutonium. In fact, nuclear weapon बनाने का जो सबसे best जो fissile material है वो plutonium है. तो यूरेनियम इनरिचमेंट का नीड बॉम्ब बनाने में भी है, एंड दूसरा ये है कि इनरिचमेंट प्रोसेस से आप प्लूटोनियम बना सकते हो. जो इनरिचमेंट प्रोसेस है दैट इंक्लूड सर्टेन टाइप ऑफ न्यूक्लियर रिएक्शंस जहां पे आप यूरेनियम को न्यूट्रॉन से बम्बार्ड कराओगे एंड दैट यूरेनियम कैन बिकम प्लूटोनियम वाया प्रोसेस ऑफ रेडियो एक्टिविटी. प्लूटोनियम, यूरेनियम बहुत सारा है दुनिया में, यूरेनियम की कमी नहीं है, बट प्लूटोनियम नहीं है. Plutonium is a very rare mineral in earth crust, does not even make sense to mine it. So whatever plutonium we make it from uranium. उससे बॉम्ब हम चाहें तो बना सकते हैं, बट हम चाहे तो रिएक्टर में भी यूज कर सकते हैं. बिकॉज ओके, तो इसका एक सिंपल सा कारण ये है, क्वेश्चन इनका ये है कि ईरान को इनरिचमेंट की क्या जरूरत है. तो जो यूरेनियम है उसके दो आइसोटोप होते हैं, यूरेनियम 235 एंड 238. जो 238 है दैट इज नॉट अ गुड फिसाइल मटेरियल, 235 इज. नाउ अनफॉर्चुनेटली जो आप यूरेनियम एक्सट्रैक्ट करोगे अर्थ के क्रस्ट से, वहां पे 99.3% जो होगा 238 होगा, 0.7% 235 होगा. दैट 0.7% composition of uranium is not going to help you in making an atomic bomb. You want uranium as pure as 80%, uranium 235. In fact, let me be more specific. You want your raw material जहाँ पे 80% uranium 235 हो, 90% uranium 235 हो, तब जाकर आप atomic bomb बना पाओगे. Reactor के लिए you need 2%, 3%. So enrichment आप reactor के लिए भी कर सकते हो, enrichment आप atomic bomb बनाने के लिए भी कर सकते हो. जो percentage है वो vary करेगा. 2, 3, 4% is good enough for reactor. 89, the moment you are entering into double digit, then you are not making a reactor. Now you are enriching it for atomic bomb. And if you have enriched 60, 70%, then you are ready to make an atomic bomb, right? टाइम लगता है, और ऐसा मान के आप चल रहे हो कि प्लूटोनियम इकट्ठा करना बहुत इजी है, राइट? आप ये मान के चल रहे हो कि जो वेस्ट है, न्यूक्लियर वेस्ट होगा, उससे प्लूटोनियम जो है इकट्ठा आराम से हो जाएगा, बॉम्ब बन जाएगा. इजी नहीं है, बहुत डिफिकल्ट है इसको करना. फास्ट ब्रीडर रिएक्टर, फास्ट ब्रीडर रिएक्टर का पर्पस है थोरियम को यूरेनियम में कन्वर्ट करना. उसमें हम यूरेनियम से प्लूटोनियम भी बना सकते हैं, दैट इज देयर. बट देखो, इंडिया जब भी 3 स्टेज प्रोग्राम की हम बात करते हैं ना, हम कभी भी नहीं लिखेंगे दैट वी आर गोइंग टू एनरिच यूरेनियम फॉर एटॉमिक बॉम्ब. अगर मान लो स्टेटमेंट ये आता है दैट वन ऑफ द कंपोनेंट ऑफ 3 स्टेज प्रोग्राम इज टू एनरिच यूरेनियम टू मेक एटॉमिक बॉम्ब, दैट्स रॉन्ग. अगर मान लो हम ये कर भी रहे हैं, विल नेवर ऑफिशियली एक्सेप्ट दिस, राइट? तो पहले तो एक ये, राइट नाउ इंडिया इज नॉट लाइक मेकिंग एटॉमिक बॉम्ब्स एज सच. लाइक वी हैव 100-120 nuclear arsenal and that is good enough, right? I guess so we'll not write that. But आपका क्वेश्चन वाजिब है. प्लूटोनियम हम यूरेनियम से ही बनाएंगे, जो इनरिचमेंट प्रोसेस है उसी का एक बाई प्रोडक्ट जो होगा वो प्लूटोनियम होगा, या जो आपका न्यूक्लियर रिएक्टर का एक बाई प्रोडक्ट जो होगा वो प्लूटोनियम होगा. ठीक है? एनी अदर क्वेश्चन? So let me like discuss few more questions which we did not discuss in the last class. And what I have done here is that in today's video description, you can find all of the 3 papers combined into one, and also all of the PPT which I showed combined into one PPT. So you can download it from there. I think superconductor वाला में डिस्कस किया था, पार्टिकल एंटी पार्टिकल डिस्कस किया था. ओके, सो फ्यू मोर क्वेश्चंस. ये 2 साल पुराना करंट अफेयर्स है, बट व्हाई आई हैव kept it here because India became a member of this organization, and hence this is an important topic. Square kilometer array observatory. क्या होगा आंसर इसका? ये जो वर्ड है ऑप्टिकल टेलीस्कोप, दैट इज रॉन्ग. इट इज नॉट. ऑप्टिकल टेलीस्कोप का मतलब होगा यू आर ट्राइंग टू गैदर विजिबल लाइट. जो स्क्वायर किलोमीटर एरे ऑब्जर्वेटरी है, दैट इज अ रेडियो टेलीस्कोप. इट इज एसेंशियली गैदरिंग रेडियो वेव्स कमिंग फ्रॉम फार अवे रीजन्स इन द यूनिवर्स. It is a collaboration among many countries. India as of now is one of those countries. And दो जगह सेटअप है इसका, एक ऑस्ट्रेलिया में है, एक साउथ अफ्रीका में है. So this much information I will remember. Radio telescope, very large area, many countries including India, and two places, one in Australia, another in South Africa. Answer here is B. C is not correct. Network of underground neutrino detector, that is not correct. Even though South Africa, Australia ठीक है, but Europe नहीं है. And it is not neutrino detection. And space-based gravitational wave, no, that is also not correct. Answer here is B. Sorry. What is the answer here? Aerogels are called frozen smoke. You should know about aerogels. They are one of the most remarkable materials which human beings have been able to make. They are often called frozen smoke because even though it's solid है, but इसके अंदर 95% air है, air which is trapped inside it. तो बहुत ही soft material है. But because air is such a bad conductor of heat, आप aerogel पे आप 500 डिग्री सेल्सियस रख दो, ये heat को allow नहीं करेगा to be conducted or radiated from one place to another place. Right? I should have a slide on it, an actual picture, an actual demonstration. You have a plasma torch, aerogel, and a flower, and that flower is not going to burn. So aerogels are known for this remarkable thermal insulation, that they do not allow conduction, convection, radiation very easily. So as a result of that, wherever you have to protect things from heat, such as rockets, spacecrafts, you are going to use aerogel. But they are very soft. So the third statement है, that they are among the strongest structural material available, not at all. They are not. They are very soft actually. थोड़ा सा आप hit करोगे, they will break down. They can do this. They are excellent thermal insulator because their nanoscale pore structure prevent transfer of heat through convection and greatly reduces conduction. This is true. This is also true, that 95% of aerogel essentially is air trapped inside that solid. So even though it is a solid material, it is called frozen smoke. But the third is not true. यहाँ पे आंसर जो होगा 1 and 2 only होगा. ठीक है? Sorry? बोल सकते हैं. बोल सकते हैं. Very unique type of colloid. Solid air suspended in solid. Air suspended in solid. This is budget. This year's budget. Budget में telescopes को लेके एक अलग से emphasis था. एक तो फोकस ये था कि जो एग्जिस्टिंग टेलीस्कोप्स हैं उनको हम कुछ अपग्रेड करेंगे, बट देयर वाज आल्सो अ बजट ऑन मेकिंग न्यू टेलीस्कोप्स. सो नेशनल लार्ज ऑप्टिकल नियर इंफ्रारेड टेलीस्कोप इज अ लार्ज ग्राउंड बेस्ड ऑप्टिकल एंड इंफ्रारेड टेलीस्कोप टू बी बिल्ट एट हानले इन लद्दाख. नाउ द ओनली थिंग विच कैन बी रॉन्ग हियर इज, बिकॉज बाकी सब कुछ तो ठीक ही लग रहा है, इंफ्रारेड ऑप्टिकल इंफ्रारेड इज फाइन, हानले, एंड दैट इज ट्रू. हानले इज वन ऑफ द बेस्ट प्लेस टू डू एस्ट्रोनॉमिकल ऑब्जर्वेशन बिकॉज ऑफ वेरी लो एयर पोल्यूशन and more importantly very low, almost non-existent light pollution, that you do not have that artificial light which essentially can obstruct light coming from very far away places in the universe. So two best places for telescopes in India, one is Hanle in Ladakh and another is Devasthal in Uttarakhand. But ये गलत है. Will be launched into space. National Large Solar Telescope will be launched into space, making it second dedicated solar observatory of India after Aditya L1. This is wrong. This is also going to be in Ladakh, but at Pangong. So Pangong Sol Lake है, ये वहाँ पे establish होगा. So answer here is one only. Because ये इस साल के budget में है, I will prepare about these telescopes, their scope, and where they will be situated. This question because of LIGO India. The government of India has in principle approved and construction also has started in Maharashtra, Hingoli district. The project is called LIGO India project. The goal of this project will be to detect gravitational wave. LIGO का जो full form है, that is Laser Interferometer Gravitational Wave Observatory. And the answer here is B. It confirmed one of the predictions of Einstein's theory of relativity, general theory of relativity, and opened entirely a new way of observing the universe. Because think about it, right now when we think about universe, we are gathering electromagnetic wave. We are gathering light. Light is nothing but electromagnetic wave. Now electromagnetic wave carries its own set of information. But now we have another way of knowing about universe, and that is gravitational wave. So gravitational waves, they were prediction of Einstein's theory of, general theory of relativity, that when you have cataclysmic events like black holes merging, or a star undergoing supernova in asymmetric manner, or neutron stars merging with a black hole. So such type of cataclysmic events, they can produce ripples in space-time itself. That space-time in itself undergoes fluctuation, and that fluctuation propagates with speed of light. And that is called gravitational wave. So gravitational wave को observe करने का एक जो सबसे famous experimental setup है, that is called LIGO. And one of that LIGO is going to be constructed in India. So that is why this question is there. Answer here is B. This is because of last year Nobel Prize in Physics, this phenomena of quantum tunneling. When we try to explain nature at very smaller scale, we realize that a lot of weird things are possible. Like weird in the sense that we will not be able to make intuitive sense of them, but they do happen. We can observe them, and we can develop our mathematics around them. We can not just observe them, we can also explain that why is it happening. One of that phenomena is called quantum tunneling. In quantum tunneling, the idea here is that if I give some particle has some energy, and there is a barrier it has to cross. So it can only cross that barrier if its energy is more than that barrier. It makes such obvious sense that you will not even think that opposite will be true. But turns out that it can happen in quantum mechanics. And no, it is not a violation of conservation of energy. If you are thinking that something has less energy and is crossing a barrier with more energy, is it a violation of conservation of energy? Not at all. Why does it happen? That's a separate question. We do not have to go into that part. But the meaning is actually quite simple. तो यहाँ पे जो आंसर है, that is, क्या होगा यहाँ पे? C, a phenomena where particle passes through an energy barrier that classical physics would predict is impossible to cross. That is true. So it happens at very smaller scale. Electrons can do that, protons can do that. Lot of things happen because of quantum tunneling. जैसे सन के अंदर जो न्यूक्लियर फ्यूजन हो रहा है, दैट इज हैपनिंग बिकॉज ऑफ क्वांटम टनलिंग. जो आपका रेडियो एक्टिविटी में जो अल्फा डिके है, दैट हैपेंस बिकॉज ऑफ क्वांटम टनलिंग. डीएनए म्यूटेशन हैपेंस बिकॉज ऑफ क्वांटम टनलिंग. इन बायोलॉजी क्वांटम टनलिंग हैज एप्लीकेशन. जो आपका एसएसडी फ्लैश ड्राइव्स में आप डेटा स्टोर करते हैं, दैट यूजेस द आइडिया ऑफ क्वांटम टनलिंग. सो लॉट ऑफ थिंग्स दे हैपन बिकॉज ऑफ क्वांटम टनलिंग. सो बट it was thought that it can only happen at smaller scale. So the scientists who got Nobel Prize last year in physics, they were able to show that if I have two superconductors, and I've talked about superconductors in the previous class, and they are connected with a very small, thin layer of insulator, then the current can pass through this thin layer even without a voltage. Because if you have insulator, current should not be able to pass through. And even if it is passing through, because it has resistance, there should be some heat production. Ohm's law, V is equal to IR. But it can be shown that this current can pass through without any heat production. So, दोनों ही बहुत अजीब से है. The first, that current can pass through, first, and second, can pass through without any type of resistance. This is a very good example of quantum tunneling. तो ये आप lab में करके देख सकते हो that billions of electrons collectively are crossing that barrier, which otherwise is not possible. So this is called macroscopic quantum tunneling. That quantum tunneling which you generally associate with small particles at very smaller scale, now it is happening at a larger scale, that you can see current happening across some barrier where it should not be possible. That is called Josephson junction, by the way. The word here is Josephson junction. Answer here is C. I wanted to mention about that last one, National Ignition Facility. First and second, they also remain in news, but third has been in news a lot in last few years, so hence we should know about this. So in America, there is a lab called Lawrence Lab. It is located in California. And they were in news because unka ek facility hai that is called National Ignition Facility. And in this facility, they were able to do nuclear fusion, and they were able to produce surplus energy using nuclear fusion. Surplus ka matlab hai they started with 100 joule, they got 200 joule. So ये पहला facility है where surplus energy using nuclear fusion was achieved. Nothing to do with neutrino detection. So that part is wrong. Ice cube is neutrino detection. So ice cube is this is a observatory located in Antarctica, deep buried in ice, and uska purpose है to detect neutrino. National Ignition Facility, that is called inertial fusion. Fermilab is another very famous lab after Large Hadron Collider, which I mentioned in the previous class. If you have to know one more very powerful particle accelerator theoretical physics lab, that will be Fermilab. So first is true, second and third they have been interchanged. Answer here is 1 only. (46) last question. क्या होगा आंसर इसका? ये पता होना चाहिए हमको. Organic matter को metal से associate कर पाना. This is called metal organic framework. So type of metallic alloy in which organic carbon based molecules are dissolved within metal matrix to improve its mechanical strength. Type of semiconductor, no. Biological framework for metal binding protein found in enzyme, no. A class of porous crystalline material in which metal ions are linked together by organic molecule with extremely large internal surface area. Answer is D, not A. Answer यहाँ पे जो है, I made this very specific because answer here is D. दो चीजें हैं. The fact that we can have organic molecules with metals associated with them, first. Second, with very large internal surface area essentially means that they can be very reactive also, because surface area plays a very important role in chemical reactivity. So these are the topics I had to discuss. The purpose of these 3 sessions were that you go and read about more about those themes. I have got those themes also uploaded. Try to read more about them. But what I said in the starting of this session, that we always have to be humble, and I have to be humble enough to accept that out of, I have told you many topics, and maybe there will be, hopefully, my hope here is that maybe some 3-4 questions will be asked by UPSC from these themes, right? Or maybe no question will be asked from these themes. But the goal here is that read more about them, but also do not let other topics which you otherwise have studied, your basic classes, your current affairs, focus upon them also, okay? If you do have queries, you can send me a mail. And for last few days, I have started this Telegram channel. So you can you can follow that also, and maybe I keep updating some important news there. So you can search this. Okay. I will see you guys around. All the best for examination. If you have any queries, send me a mail. Okay? And all the best, guys.
The issue with most UPSC aspirants is not effort, it is foundation. A strong foundation means 3 things. Learn in the right sequence so subjects reinforce each other. Apply early through answer writing and testing. Correct continuously through proper diagnosis and feedback. This is what the GS Foundation course at Vision IAS is built around. Connected GS coverage, not isolated subjects. Early writing and testing, not last-minute panic. Diagnostic test analysis with clear corrective steps. Current affairs organized into usable themes for prelims and mains. Regular mentorship to prevent drift. Effort is common, structure is rare. If you are serious about UPSC, start by getting the foundation right. Upcoming classroom batches: Karol Bagh 9 March 11 am, 13 March 8 am and 24th March 5 pm. And GTB Nagar batches for English 17 March 8 am, 25 March 6 pm. And for Hindi 16 March 11 am. For more details, visit our website or call us on the given numbers.
यूपीएससी 2027 का लक्ष्य जितना बड़ा है उसे पाने की योजना भी उतनी ही स्पष्ट और संगठित होनी चाहिए। विजन आईएएस लेकर आया है क्लासरूम फाउंडेशन कोर्स 2027 के 12 से 14 महीनों का एक ऐसा सुव्यवस्थित कार्यक्रम है जहां आपकी तैयारी एक अनुशासित और वैज्ञानिक ढांचे में आगे बढ़ती है। यहाँ क्लासरूम लर्निंग के साथ साथ आपको मिलता है लाइव और रिकॉर्डेड सेशंस का विकल्प, जिसे आप अपने व्यक्तिगत ऑनलाइन पोर्टल के माध्यम से कभी भी और कहीं भी एक्सेस कर सकते हैं। तैयारी के सफर को और भी प्रभावी बनाती है हमारी व्यक्तिगत क्लासरूम मेंटरिंग। हमारे एक्सपर्ट मेंटर्स नियमित मार्गदर्शन और स्मार्ट स्टडी मैनेजमेंट के जरिए आपकी तैयारी को एक सही दिशा और गति प्रदान करते हैं। शैक्षणिक दृष्टिकोण से देखें तो यह कोर्स मुख्य परीक्षा के सामान्य अध्ययन के चारों प्रश्न पत्रों, सीसेट और निबंध का व्यापक कवरेज सुनिश्चित करता है। विषयों की समसामयिक समझ विकसित करने के लिए मासिक करंट अफेयर्स मैगजीन और रिवीजन क्लासेस के साथ साथ पीटी 365 और मेंस 365 की विशेष कक्षाएं भी इस कार्यक्रम का अनिवार्य हिस्सा हैं, ताकि स्टैटिक और करंट अफेयर्स के बीच एक सटीक संतुलन बना रहे। तैयारी की गहराई मापने के लिए निरंतर अभ्यास अनिवार्य है, इसलिए डेली असाइनमेंट और साप्ताहिक मिनी टेस्ट के साथ साथ मासिक प्रीलिम्स और मेंस टेस्ट के माध्यम से आपकी प्रगति का निरंतर आकलन किया जाता है। इस कार्यक्रम की शक्ति है हमारी प्रतिष्ठित ऑल इंडिया टेस्ट सीरीज, जिसमें प्रीलिम्स के 35, मेंस के 30, सीसेट के 25 और निबंध के 5 टेस्ट का समावेश है। साथ ही संधान पोर्टल पर 25,000 से अधिक प्रश्नों का विशाल संग्रह आपके अभ्यास और विश्लेषण को और भी सुदृढ़ बनाता है। अब समय है अपनी मेहनत को एक मजबूत, संरचित और निरंतर प्रक्रिया में ढालने का। विजन आईएएस के साथ जुड़े और यूपीएससी 2027 की अपनी सफलता की राह को एक स्पष्ट दिशा दें।
Prelims is in 3 months. This is where preparation shifts, not from studying to stopping, but from expanding to consolidating. At this stage, what matters most is strengthening core areas, fixing weak zones, analyzing mistakes deeply, stabilizing mock scores, refining your attempt strategy. That is exactly where the Lakshya Prelims Mentoring Program plays a decisive role, offering structured direction, performance correction, and continuous guidance when execution matters the most. Starting from 23rd February, Lakshya supports this critical phase with a focused revision roadmap, personalized tests and competitive quizzes, 25,000 plus curated Sandhan questions, data-driven performance analytics, mentor guidance till the exam. Lakshya is not about studying more, it's about executing better with expert guidance. To know more or register, visit our website or call us on the given numbers. Are you ready to face prelims head-on? Because prelims is not just about knowledge, it's about performance under pressure. That's where Abhyas, the All India Prelims, GS plus CSAT mock test comes in. Designed as a true exam simulation, Abhyas recreates the real UPSC. Prelims experience the same pattern, the same time constraints and the same intensity. It places you in a national arena, competing with aspirants across India, backed by an all-India ranking that reveals your true standing. Attempted by 1 in 3 top 50 rankers, Abhyas has become a trusted benchmark for serious aspirants. With detailed post-test analysis, live discussions and complete syllabus coverage in both English and Hindi, it ensures your preparation is not just extensive but exam-ready. 3 full-length offline simulations will be conducted across 100 plus cities on 5th April, 19th April and 10th May. From 9:30 am to 11:30 am for General Studies, that is paper 1, and 2:30 pm to 4:30 pm for CSAT, that's paper 2. Challenge your preparation, test your performance. For more information or register, visit our website or call us on the given numbers.
All serious aspirants are able to solve polity, economy and geography questions in the prelims. But when it comes to ancient and medieval history and art and culture, everyone stands on the same platform. Whether you are a fresher or a repeater, attempts don't matter here. Most aspirants end up guessing answers, relying on vague recall and finally getting stuck in a dicey zone near the cutoff. And it is exactly here where margins matter, that marks are lost or gained. To create a clear edge over the cutoff, Vision IAS Prelims Speed Up is for you. The real challenge with history and culture is not effort but direction. Multiple sources, overlapping facts, confusing timelines and factual overload make these subjects risky. UPSC further complicates things by framing indirect elimination-based questions where partial knowledge often backfires. As a result, even well-prepared aspirants hesitate at crucial moments. Prelims Speeder is designed as a 20-day last-mile score booster to solve this exact problem. Instead of broad coverage, the program focuses only on high-grade areas that UPSC repeatedly asks in ancient history, medieval history, and art and culture. The emphasis is on conceptual clarity, chronology, and intelligent linkage, so facts finally start making sense together. With concept-driven classes, the course tells you what to remember, what to ignore, and how to apply. Structured daily practice and tests train your mind for elimination, helping you avoid traps and guess less. The Smart Source consolidated content removes the burden of multiple books and speeds up the revision. In just 20 focus classes for ancient and medieval history and art and culture, Prelims Speed Up converts static confusion into scoring confidence. If you want to move from cut-off anxiety to cut-off safety in Prelims 2026, this program gives you that decisive edge. Join today to get this edge.